Welcome everyone for this Perform Colloquium. So it's our pleasure to have with us Professor Edith Hamel, Director of the Laboratory of Cerebrovascular Research from the Montreal Neurological Institute at McGill. Dr. Hamel is notably an expert in when studying the concept of neurovascular coupling. I know some of you might have done fMRI study. You should remember that when you do fMRI, all what you're doing is based on one model of the neurovascular coupling. So I think this is very important for all of us in that field. So today, Dr. Hamel will talk to us about vascular cognitive impairment and dementia, white matter alterations, and physical exercise. Also the link with physical activity, which is of course of great importance for us at Perform. So it's a pleasure to have you, Edith, and we're looking forward to your talk.
Okay, well, thank you for the invitation, and thank you particularly to you, Christophe, and also to Claudine, who invited me. I think both of you invited me in for a spring seminar that unfortunately could not be happening due to COVID-19. So thanks for getting back to me for a fall presentation, a virtual presentation. So thanks everybody for being there. I will try to show you a little bit of the work we've been doing with VCID and as well with exercise. And as you will see, it's something I never thought I would be doing research on exercise. So you will see that I could not do it alone, and I would explain to you why. So this is a early morning this summer. So as you can see, it was nice to see the sun getting up at the time that I was trying to get up some mornings. So a bit of the outline of my talk. So I would like to introduce what is VCID, because it used to be called vascular dementia, and now we refer to it as VCID. Also, I would like to introduce our animal model, because there are many models, but we selected one which I think has advantages. No model is perfect, but at least we have to know what we can do with the model. I will be talking about the cerebrovascular and cognitive outcomes of the model, and then we looked for pathological biomarkers that we could identify. And that is important because if you want to look at pathology and at reversal of pathology with therapeutical approach, I think we have to be able to have markers that can direct to us that we are actually improving something. And therapeutical approach, again, as I said, I will be talking about exercise, but it was very difficult for me having never done exercise before, and it was a kind of a very obscure type of concept in terms of pharmacology. So I said, we will do exercise, but we will do a comparison, head-to-head comparison with pharmacotherapy. And I think we learned quite a lot of interesting things, and I'm happy to share that with you. And the paper was just recently published actually in GLIA a few months ago. So if we talk about VCID, which used to be, as I said, commonly referred to as vascular dementia, it is actually the dementia or the loss of cognitive performance that experience people mainly due to various types of vascular problems. So it can be due to cardiac disease, it can be due to ischemic, hypoxic event, it can also be due to small hemorrhagic diseases. And as well, a lot of emphasis has been coming out over the last years with those hereditary small vessel disease, namely CADASIL and CARASIL, so the dominant form and the recessive form of small vessel disease. And what is interesting in that is that when you look at MRI and you do a structural MRI, depending, yes, if it's a massive post-stroke or if it is a very strategic small infarct, you will see very localized images on the MRI. But most of the time, it will be targeting the white matter. So this is a kind of a landmark that in terms of VCID, a lot of the pathology in terms of structural changes in the brain can be seen by these hyperintensities and white matter lesions. So all of that is actually believed because the causes of VCID is really vascular, that cerebrovascular disease, chronic hypoperfusion, so chronic loss of oxygen and of glucose supply, will actually lead to all of these cascade of events that will lead to cognitive impairment. So that has been for many years, it has been an issue on how can we actually try to study VCID in animal models. So there has been lots of models, and I think the most studied one and probably the most, or the original one, is by inducing chronic cerebral hypoperfusion by doing bilateral carotid artery stenosis. So I think that model has been teaching us a lot of information. It is producing chronic cerebral hypoperfusion, but I think one of the drawbacks is that sometimes if you are not a real expert in that model, when you do the surgery, you get a very abrupt decrease in cerebral blood flow, sometimes very severe. And then the blood flow will recover and will gradually stabilize to about minus 20–25% of perfusion. So really chronic hypoperfusion. But that first acute drop, can sometimes lead to neuronal death in the hippocampus. And it has been one of the problems, even though it's a model, as I said, that we learn a lot from. There is also the endothelin-1 induced ischemia, where you do multiple small injection of endothelin-1 in the cortex or in other area. And since endothelin-1 is a very potent vasoconstrictor, then you produce local infarct, multiple local infarcts. And that also has been a very interesting model. A bit similar to inhibition of endothelial nitric oxide synthase, because by decreasing the synthesis of NO, which is a potent vasodilator, then the vessel get into a constricting mode and you have basically less perfusion to the brain. And there has been, over the recent years, the transgenic mouse model of CADASIL, which is a Notch3 mutation. And that one has been used a lot by the group of Anjoutel in France, in Paris. And they have done beautiful work. And they have actually won the Brain 2019 prizes with that work. And I think one problem with the model is that it still doesn't show cognitive deficit, likely due to the background or the strain of the mice that they have used. So they're now working to change the strain so that they will have more than the vascular pathology, but also be able to basically reproduce a cognitive deficit. So our model actually was based on a combination of cerebrovascular pathology, TGF mice, and I'll come back to that, to which we added a risk factor, namely hypercholesterolemia. So why did we do that? Well, we did that because we had been working for many years with the TGF mice, which is a model of over a transgenic mouse model that overexpressed transforming growth factor beta 1. It's a model that was developed many years ago by Tony Weiscory and Leonard Muke, and it actually mimics the vascular pathology that you can see in the brain of Alzheimer's disease and of VCID patient or vascular dementia patient, but really only the vascular pathology. And as the research went on and on, it actually has more and more relevance to actually the genetic form of small vessel disease like CADASIL and CARASIL. And why am I saying that? Because if we look at the CADASIL, so CADASIL is a pathology where there is basically a Notch3 mutation. And that actually results in all the cascade of events that will lead to the deposition of gum, which are those proteins deposited around the vessels in the brain of the patient with CADASIL, and all the vascular pathology as well as the neuronal pathology. So that is CADASIL is a Notch3 original pathology. Then in the CADASIL, so CARASIL, the recessive form, it is really this protein here, the HTRA1, or the high temperature requirement protein A1, which is also mutated, and it's a loss of function. So by losing function, you actually enhance or promote TGF-beta 1 signaling, which actually will result in vascular fibrosis, accumulation of extracellular matrix protein, vascular contraction. So basically what we see in the transgenic TGF mice, which actually overexpress TGF-beta 1. So this is why in that sense, we thought that it could reproduce quite well some aspect of CARASIL, but it became even more relevant, I would say, over the last couple of years when it was found by a group in Germany that actually the loss of function of the HTRA1 protein is also present in CADASIL patients. So there is clearly a link between small vessel disease that lead actually to cognitive impairment and with the signaling of TGF-beta 1. So I think the model is interesting in that respect. So what is the model actually? So as I said, it's a model that was developed originally by Tony Weiscory and Leonard Muke to mimic the vascular pathology that can be found in Alzheimer's disease and in vascular cognitive impairment and dementia. So it is an overproduction of TGF, an active form of TGF-beta that is present, that is done through the astrocyte. So there is a only brain selective overexpression of TGF-beta 1. And what is interesting in the model is that it really reproduces quite well the fibrosis of the vessel. So as you can see here, you have a wild type animal, and in the TGF mice, you have accumulation of protein in the extracellular matrix of the vessels. So the vessels are stiff, they're rigid, they are thickened, vascular fibrosis, so very, very clear in the model. They also have an increase in string vessels. So string vessels are actually degenerating capillaries where you do not have a lumen anymore, you do not have the endothelial cell. It is just a connection of the extracellular matrix that connects between capillaries. And what is interesting is that these string vessels or degenerating capillaries are actually thought that we can regrow endothelial cell into those vessels and make them function again. So there is a lot of interest into that pathology of the string vessel pathology, which is present in Alzheimer's disease and in VCID. There is also small cerebral hemorrhage in the TGF mice, which is very relevant to VCID. And as you can see, however, there is no amyloid beta pathology. So it is not a model of amyloid pathology. There is no CAA around the vessel. There is no amyloid beta induced oxidative stress. And there is no cholinergic denervation, which is another landmark of Alzheimer's disease. So clearly it's a model totally independent of the amyloid pathology and the neuronal pathology of Alzheimer's disease, but it is mimicking the vascular pathology. It has vascular, cerebrovascular, and inflammation. So if we look here at the perivascular inflammation, we see activation of the astrocyte, activation of the microglial cell, clearly enhanced into the TGF mice. There is impaired cerebrovascular reactivity, so they lose endothelial function, so they are more prone to vasoconstriction, again, another marker of VCID. And they have chronic hypoperfusion. And that chronic hypoperfusion is actually quite regular. It's about 20, 25, 30% maybe at most, but it's very regular. So I think that's a good model of chronic hypoperfusion with no initial massive drop. What is interesting is that they don't have these mice, cognitive impairment or very, very late. So we tested mice up to 26 months sometime, and we could not detect cognitive impairment. In other cohort at about a year or so, we could see a little bit that they would be moving away from the wild type, but they don't really have strong cognitive impairment. So this is why we thought they have a very good vascular pathology. Then we said, okay, when we talk about Alzheimer's disease and VCID, the main risk factors are aging. The primary one is aging. And then all of those cardiovascular disease are actually the main risk factor for both Alzheimer's and VCID. So we said, what about if we actually add on to the TGF mice that had this vascular pathology with a chronic hypoperfusion, which is not very severe, but we add on a risk factor? What will happen? So we decided to use hypercholesterolemia. So we added to the diet 2% cholesterol, and we treated the mice for 3 to 5 months, and we treated them up to the time that they were what I would call a young adult, 6 months or so, and an aged adult, 12 months around, and maybe a bit above that. And it is really the work of Ching Kan Tong. He initiated that work, and then Liane took over for a PhD project. Actually, Liane now graduated a few months ago. She's now at Cornell University doing beautiful work with 2-photon microscopy and genetic manipulation. And she is actually a student who came to me from Concordia University. And so the question we asked was, will cerebrovascular pathology in TGF mice be worse once they get hypercholesterolemic? And will we be able to actually trigger cognitive impairment in these mice following high cholesterol diet? So we measured cholesterol in the blood, and as you can see in both adult and age animal, whether they were wild type or TGF mice, we were able to get a clear increase in the level of total cholesterol in the blood. Now, we also measured cholesterol in the brain tissue, and we did not see an increase in cholesterol in the brain. However, this is kind of expected because the brain is synthesizing its own cholesterol through the astrocyte. And we actually have metabolites from the cholesterol in the blood that can access the brain, like the oxidized form of cholesterol, but we did not measure that. So it is possible that we had some of the metabolites of cholesterol reaching the brain, but we did not measure them in our tissue sample. So I'm going to be studying about, Christophe made reference to neurovascular coupling, which is the basis, obviously, of fMRI bold signal. So in the rodent, the neurovascular coupling is classically studied by doing whisker stimulation, which increases blood flow in the visual cortex or the sensory cortex of the whiskers. And as you can see, if we look at the bar graph, the wild-type mice, this is the type of CBF response that we get, and TGF mice are already impaired in that, even as adult or even as more age adult. And when we treated them with high cholesterol, we did not actually worsen this already impaired response. And you can see here the curves. This is the stimulation. We have an increase in blood flow in the wild-type animal. The response is smaller in TGF mice, and it is not actually worse in the mice fed with a high cholesterol diet. Then we looked at vascular reactivity, because that's also something quite important. And TGF mice are known to have impaired endothelial function primarily, and they are more into a mode of vasoconstriction. So their vessels are primarily in a vasocontractile mode. So in order to do that, we did that in vitro, where we can isolate the middle cerebral artery or the posterior cerebral artery of the cerebral blood vessel. We mount them in vitro into a pressurized system. So they are mounted between two micropipettes. And we're talking vessels here that do about, in diameters, 80 micron diameter. So it is really not something that is so easy to do. And then we can superfuse the chamber, the medium in the chamber, by specific drugs. And as I'm showing here acetylcholine, which is a typical endothelial dependent dilator. And in the adult mice, you can see that the TGF mice, actually the black triangle, are already impaired for the acetylcholine response, and they are also impaired for the calcitonin gene-related peptide response, which is a peptide that acts also through the endothelial cell, although there might be a small component at the level of the smooth muscle. And as you can see, these two responses are massively altered in TGF mice. They were not worsened in the TGF mice that were fed a cholesterol diet, but we can see, interestingly, that in the wild-type mice that were fed a high cholesterol, even as adults, they were starting to be less effective in dilating to an endothelium-dependent dilator like acetylcholine. When we go to more age animals, so in the aged TGF mice, we can see that they were impaired, but that impairment became significantly worse, as was the case for the CGRP response. And in that case as well, the wild-type mice became very significantly impaired. So that is telling us, and maybe me as a probably adult of a certain, not a young adult anymore, I am most likely more susceptible to high cholesterol food than the younger adult or maybe the student attending that talk. Maybe you can sustain better a high cholesterol diet or junk food than we can at a certain time. So maybe you should take advantage of that while it is still time, because clearly, as we can see, it is affecting the vasculature of your brain. Now, if we look at the behavior, so we did multiple tests. We did first the Morris water maze, and that's the classic Morris water maze that we use in the lab, where we have 3 days of habituation, so where the mice are familiarized with the room and with the pool and everything. Then we remove we put into the water the platform, and then the mice have 5 days to get used to find the platform. And then we remove the platform and we perform the memory or the probe trial. So as you can see, whether it is adult or age TGF mice, it's the only group that actually had deficit in learning the task. Even the wild-type mice were able here, the blue circles, and here the blue circle, even under high cholesterol, the wild-type, whether they were adult, young adult, or age adult, they were still able to learn the task quite well. And when we look at the memory in the probe trial, again, only the TGF mice were actually impaired when they were having high cholesterol diet. So clearly the high cholesterol diet was able to trigger cognitive impairment selectively in the TGF mice when we're talking here about spatial memory. Then we look at executive memory, which is more commonly affected in vascular dementia. And here we can see, and I think those mice were about 8 months of age. This is the novel object recognition test, where the mice is presented with 2 similar objects, then we change one object, and normally the mice will spend more time looking at the novel object. And as you can see here in these mice, both the wild type and the TGF high cholesterol mice were impaired in that executive function. And that is kind of interesting. Then we perform another task, which is the Y-maze alternation. And that's a mix of spatial memory and executive function again. And as you can see, even though there was a trend for the wild-type high cholesterol mice to basically perform a little bit less than the wild-type, it was significant only in the high cholesterol TGF mice. So they were more susceptible to cognitive failure, the TGF mice, when we were adding this high cholesterol as a second factor for dementia. Then we looked for markers, because it's nice to see that we do have cognitive impairment. It's nice to see that maybe we can actually worsen vascular reactivity, but in the brain, can we see something that will actually be a good marker of the cognitive failure that is happening in these animals? So we look at many things, and I'm not going to show everything, but I think I just want to show a few that we used later on when we did the pharmacotherapy and when we did the exercise therapy. So something that was kind of interesting and that we had to look at, because we know that the TGF mice, as you see it here with the activated astrocyte labeled with GFAP, TGF mice already have activated astrocyte or astrogliosis in their brain, as can be quantified here, and it was not basically worsened following high cholesterol diet. So both the intensity and the area positive for GFAP were comparable between the TGF and TGF high cholesterol, and it was not affected in the wild-type animal. However, looking at the microglia labeled here with IBA1, as you can see, TGF mice already have bigger microglial cells, so the cells are activated, and that is clearly quantified here. But we can see that the wild-type fed a high cholesterol diet already started also to have reactive microglial cells. And for the TGF mice, the high cholesterol diet did not worsen already the activated microglial cell. And that was true in the gray matter. And then, as I told you at the beginning, that the white matter is what is mainly affected in VCID. Then we looked in the white matter, and this is the corpus callosum. So as you can see here, delineated, and we looked for activated astrocyte. So again, wild type. We can see that the TGF mice already show sign of activated astrocyte, and it was not worsened by the high cholesterol diet. When we look at the microglial cell, again here in the corpus callosum, again we find that the TGF mice have bigger microglial cells, so they are activated. And in that case, though, in the white matter, the wild type did not show actually an increase in the microglial activation. And again, in the TGF mice, high cholesterol did not worsen that phenotype. Okay, so then we looked at the white matter, and then we looked at a specific type of microglial cell that are labeled with galectin-3. So galectin-3 is a lectin that is found actually in microglial cells and even in macrophage that are found selectively in the white matter and that are known to phagocyte white matter debris when there is white matter dysfunction. And we had found a lot of interesting paper that had found correlation between the appearance of galectin-3 microglial cells in the brain together with the development of cognitive impairment. And this was true in models like the Cuprizone model of demyelination. Wherever there was actually cognitive deficit, these microglial cells were actually being detected, as well as in some models of primate, non-human primate with cognitive failure that were showing galectin-3 correlation with actually these deficits. So as you can see here in the internal capsule, we can see the wild type high cholesterol. We can see that TGF mice already have some of those galectin-3 positive cells, but whenever they're fed a high cholesterol diet, it's like a Christmas tree. It is shining, the cells are very bright, very, very intensely labeled, as can be shown. And that was also seen when we look in the corpus callosum, the same phenomenon. TGF mice do have a little bit of these positive galectin-3 cells, but when they are fed a high cholesterol, it was much more prominent. And that was clearly very, very clear in all the animals that we looked at. Then we asked a question, as I said, because these galectin-3 cells can be either microglial cell or they could be invading macrophage coming from the periphery and infiltrating the brain. Since we know that there might be small hemorrhage, maybe the blood-brain barrier could be a little bit impacted in the TGF model with high cholesterol, Then we did a collaboration with Dr. Sam David at the Montreal General Hospital, trying to see if we could find markers that would indicate to us that they were invading cells from the periphery. And by looking at all the markers, CD45, CD3, LY6C, all of those would be actually giving us indication that it would be monocytes, leukocyte, or macrophage, or T cells that could be found into the tissue. So, and all of these analysis of FACS sorting were done in basically isolated white matter from the mice, TGF mice with high cholesterol. And here you have all the groups, and I'm not going to talk about the exercise at this point, but as you can see, there was no change into any of these markers, telling us that the changes that we see in the galectin-3 positive cells are really from the resident microglial cell, and it is then the cells of the brain that are being activated. Looking at these are the string vessels. So if I'm showing this one, those are two capillaries, and this is a string vessel in between. And as you can see, they exist into the brain. They exist into all the brains, but they are more numerous into the brain of TGF mice. So we looked into the cortex, into the hippocampus, and into the white matter. So when we look into the cortex, we could not see a significant changes in TGF mice, whether fed or not a high cholesterol diet. However, in the hippocampus, we can see that TGF mice already have more of these vessels, and this is further increased when the mice are fed a high cholesterol diet. In the corpus callosum, both were already quite high in the TGF mice, not getting worse with high cholesterol, and significantly different from the wild type. And the same in the white matter internal capsule. It is clearly worse in the TGF mice compared to wild type, and it was a little bit worse when they were fed high cholesterol. So clearly white matter is affected by high cholesterol diet, even more than it is in the TGF mice. So maybe that's why TGF mice can at some point develop cognitive failure, but maybe the pathology is not strong enough to really make them collapse and have cognitive impairment. But we don't need too much, it seems, to make them lose that reserve of whatever it is. So if I summarize rapidly that first part. So clearly the high cholesterol diet is able to worsen cerebrovascular reactivity in an age-dependent manner, and it even affected wild type. However, it is not impairing neurovascular coupling in the TGF mice because it's already impaired. It is able to induce cognitive deficit in both, actually, sometimes depending on the test, wild type and TGF mice, but the TGF mice are actually more sensitive. They were impaired in all the tests that we've done. The high cholesterol diet had no worsening effect on astroglial cell and microglial cell, but in the white matter, yes, they were able to trigger that selective increase in the galectin-3 positive microglial cell. And they were also able to, the high cholesterol diet was able to worsen the string vessel pathology in specific region. So that really became, the galectin-3 then really became one of the best marker that we could see, strikingly, and that was also reported in the literature to actually be appearing in the white matter in different model with cognitive impairment. So then, treatment. So knowing what high cholesterol does to the model, knowing that we do have some markers that we can look at, then we decided to look at, as I said, pharmacotherapy, and more than pharmacotherapy, also exercise. So why did we select simvastatin? Because as you can see, this is the pathway for synthesizing cholesterol. And when we gave simvastatin, it is a blocker of that MG coenzyme reductase. So the first enzyme in the pathway to synthesize cholesterol, this is where simvastatin is acting. But we are giving the mice cholesterol, so we don't really care about blocking synthesis of cholesterol because we are feeding the mice cholesterol. But we selected the simvastatin because it was shown to have benefits on cognition in man. It was shown to rescue memory in multiple models of Alzheimer's disease mice. And we had used it in TGF mice to actually rescue the vascular pathology of the TGF mice because it was quite effective. And all of these effects are actually independent of the cholesterol lowering effect of simvastatin. They are due to its pleiotropic effect, being an antioxidant, anti-inflammatory, pro-angiogenesis, pro-neurogenesis. So it is actually a dirty drug. in a sense, but these effects can be quite important in its efficacy. And then the second drug that we use, which is even a more dirty drug, if I can say, is exercise. And we had the mice spinning. So the first part of the study, we were very naive to exercise, as I said. So we put the mice with the spinning wheels, and all the wheels were actually equipped with a counter. So we could be counting how much kilometers they were actually doing overnight. And we realized that the mice were actually addicted to the wheels. So they could actually travel about more than 20 to 25 kilometers sometime overnight. So that was clearly not translational. And the mice were getting exhausted. So after 1 month, we shifted them to 2 hours a night for 5 weeks or 2 months, depending on the treatment. And then in the last series that we've done of studies, we did 3 hours a night for as many months as we wanted. Because at that time, in the limited exercise, the mice steadily over the treatment period were actually running about 3 to 4 kilometers a night. So which is totally translational, because we did the mathematics in terms of the, the dynamics of the human and the mouse and the exercise that they were doing. It would actually correspond to about 40 to 45 fast walk or slow jog in a human, which we think can be translational. So now when we look at the effect, so I'm going to show you the classic drug effect that we could see in many of these studies that we've done. So looking at the neurovascular coupling, as you can see, and I'm only showing here the TGF mice. TGF mice we know are impaired, and they were not more impaired by high cholesterol, but simvastatin was able to improve that significantly, bringing it to the level of the TGF mice, which is still not the level of the wild type mice. But in the study done here with the exercise mice, and this is limited exercise, you can see, interestingly, that even the wild type on high cholesterol, as I mentioned, had actually a decrease in the neurovascular coupling response, and that was fully normalized by exercise. The TGF mice here, whether they had high cholesterol or not, were impaired, but they were exercise fully normalized that response. So exercise very potently was able to normalize neurovascular coupling response in wild type and TGF mice. When we look at the in vitro vessel reactivity, so the triangle here are the TGF mice, whether they are black or white triangle, mean that they are either fed or not a high cholesterol diet, and we know that they have been impaired. But when they are treated with simvastatin, they fully recover the ability to dilate to acetylcholine, the ability to dilate as well to CGRP, as shown here with the open square. And when we do exercise, and I'm only showing the most important group here, so the TGF mice are impaired in the acetylcholine, whether treated or not with high cholesterol, and they are fully rescued by exercise. And that was the same with CGRP here. They were impaired with high cholesterol or not, and fully normalized with exercise. So both, basically, simvastatin and exercise are able to rescue cerebrovascular function quite effectively, and more so, I would say, exercise, because it normalized even to the level of the wild type animal, which was not the case with simvastatin. The wild type would be at about 25 to 30, as shown here. We also did baseline blood flow, because we know that there is chronic hypoperfusion in the model that we're using. So we did iodo and tipirine radioautography, and you can see wild type here in TGF, so clearly chronic hypoperfusion. When they're treated with a high cholesterol diet, there is even a further decrease in the baseline cerebral blood flow. And when the mice here were actually exercising, you can see that there is a basically recovery into the baseline blood flow. So we didn't do simvastatin in that group, we only did the exercise. And as you can see, even though the trend is pretty clear into TGF high cholesterol following exercise recovery, and maybe the same pattern here in the wild type animal, I did not put quantification here because that was a nightmare study, the one study that you don't want to give to a student if you don't want them to get depressed, because what we realized is that when we treat the mice with high cholesterol, the blood became quite viscous, so we could barely not get the blood that we needed to quantify the radioautography in the blood. So the brain was okay, but we could not quantify basically as we would have liked with the correction for the blood amount of radioactivity. So this is why we show the result but we cannot really give you a statistical confidence in that. But I think the brain showed quite convincing data. So then in the recovery for the cognitive deficit, so this is our TGF mice with high cholesterol again, the one that showed the learning deficit. So when we actually had them treated with simvastatin in blue, you can see that they behave beautifully, that they learn the task as well as the wild-type animal. And then when we did the probe trial, the TGF mice were the one that were impaired, and the one treated with simvastatin were comparable to TGF mice. So again, simvastatin was again in that test quite powerful. When we did exercise, so in the NOR test with exercise, as you can see in that study, TGF were not impaired, high cholesterol, they were impaired, small improvement with simvastatin, but it was not significant, but exercise was significant, bringing them to basically the level of the wild-type animal. So that was free exercise for 1 month. When we did the study with limited exercise with all the groups of animal, as you can see, there was impairment in the wild type, fully recovered by exercise, and the same here into the TGF high cholesterol diet, full recovery by exercise. And when we look at the white maze, where only the TGF mice were significantly impaired, we could see that exercise was able to fully recover. So basically, in all the tests that we've done, exercise was very powerful in recovering the cognitive deficit that we saw. So then we wanted to look at some of the markers that we had looked at. So what about the brain pathology? So as I said, in the gray matter, we know that TGF mice have an astrogliosis. We know that it's not worse by high cholesterol diet. As you can see here, it was not really removed by simvastatin, but exercise was very powerful in being able actually to resolve that, as quantified here. So basically, simvastatin was not effective, but exercise brought it almost to the level of the wild-type animal. And when we look at the microglial cell into the gray matter as well, you can see that the TGF mice have basically swollen cell bodies. It was not really worse in the high cholesterol. Simvastatin didn't do too much, but the exercise really was able to normalize. As you can see here, significant reduction, almost to the level of the wild-type animal. And when we did the study with all the groups again, you can see that exercise was very potent in normalizing the size of the microglial cell. What about our GAL3 marker, which was very, very, very intense into the high cholesterol diet mice, into the internal capsule? So as you can see here with simvastatin, we were able to decrease that marker, as shown here in the quantification with all the group. So there was a decrease. It was not back to the TGF level, but it was significantly decreased from the high cholesterol diet. But exercise was even more potent, as you can see in that specific animal. Basically, there was almost nothing left. So very, very, again, for both simvastatin and exercise, they were able to basically remove that reaction of the GAL3 positive microglial cell. And that was shown also in the corpus callosum. As you can see with simvastatin again, it decreased into the level of the GAL3, not as low as the TGF mice, but significantly less so than the high cholesterol mice. And that was even the same for the exercise animal. It was even more effective in reducing the GAL3 inflammatory response in the white matter. String vessel pathology. So as we've seen, it was sometime worsened, sometime not, depending on the region. But what was quite interesting, and that's both simvastatin was very potent in reducing the string vessel into the corpus callosum and into the internal capsule, as shown here. And here I am showing exercise into the corpus callosum, very effective. So both also able to reduce the string vessel pathology very effectively. Then we ask the question, we have less inflammation, as actually we labeled with galectin-3, which we know is a microglial cell that contains this lectin that normally these cells are there to repair myelin and to remove the debris of myelin. We see that we have less capillaries degenerating by the treatment, so we believe more perfusion to the white matter. as was shown in the slide where I had this, baseline blood flow. We know that exercise was able to improve that, and it was in both actually gray and white matter. So then we asked, what about function? What's happening with the function of the white matter? So then we collaborated with a group of Michael Feeling in Toronto, where they are able to isolate the corpus callosum, and then they can record by stimulating at one side. They are able to record at the other side, and they actually measure the first peak, which corresponds to the myelinated axon, and the P2, which is the unmyelinated axon. And then what we noticed there is that there was no change in conductivity across the myelinated or the unmyelinated axon as a whole, basically. But when we actually did different intensity stimulus, going from a very physiological to sometimes a plateau, we could see that there was, in the high cholesterol treated group, a deficit into their response in the amplitude of the P1. And as you can see here in the green triangle, that was fully normalized in the mice that exercised by the green, very bright green triangle. And that impairment was not seen into the P2. So that means that in the exercise group, it seems that exercise was able to actually help functioning or transmission of the propagation of the signal into the white matter at specific stimulus intensity. So then we were curious to know, knowing that when we talk about white matter, when we talk about preserving white matter, preserving myelin, we have to have oligodendrocyte. We have to have those that do the myelin. And there is in the supraventricular zone, where basically neurogenesis and oligogenesis is happening, it says always refer to the vascular niche. So in order to be able to, for neurogenesis to be happening, they need to have a healthy vascular niche. And we know that the vessels are actually sick in our model. So we try to see, is that going to impact on neurogenesis or oligogenesis in that case? So we decided to look at different markers into the oligogenesis, one being a very, very early marker, which is basically a very early precursor cell, the PSA-NCAM. And then we looked into the precursor oligodendrocyte precursor cell to the mature oligodendrocyte. We decided to look at OLIG2, and which is across the whole spectrum of the oligodendrocyte can be detected. And then CC1 that can be seen into the mature oligodendrocyte, then together that the PS1-NCAM, PSA-NCAM. So this is in that oligovascular niche in the periventricular zone where you see PSA-NCAM in the wild type high cholesterol, decrease in TGF, decrease in TGF high cholesterol to the same extent. But when the mice were treated with simvastatin, we actually reinstated the amount of that marker in those cells in this supraventricular zone. Then we look at the OLIG2 in the corpus callosum, and as you can see, there was a trend to decrease in the high cholesterol treated animals, a trend to be normalized by simvastatin, but nothing was significant except the decrease into the TGF mice. In the exercise group, however, and as you can see here for OLIG2, again in the corpus callosum, you can see that there was a decrease significant in the wild type, significant in the TGF mice, and that it was significantly recovered by exercise in the TGF with the trend in the wild type animal. And then when we looked at the CC1 colocalization with OLIG2, we could see clearly an increase in the matured oligodendrocyte in the exercise group. So exercise is helping basically, or is favoring maturation of oligodendrocytes. So this is a summary of that basically second part. And I think the only thing I would really like to insist upon is that exercise, but not simvastatin, reduce astrogliosis and microgliosis in the gray matter. But beside that, they were both able to improve the GAL3 signal or reduce GAL3 signal in the white matter. They were both able to basically rescue cognitive deficit, to rescue cerebrovascular deficit. So they were actually, and they actually, for exercise, it rescued the transmission in the white matter. So I think somehow both of those drugs were actually quite effective in rescuing not only the pathology that we saw into the cognition, but as well into the brain. So then I'd like to close in with that last two more slides. We need to understand what's happening in the white matter. in order to confirm if it is really related to cognitive deficit. So what we've done there, we have another model, and I just want to present it to open up the question. This is an inducible model with tamoxifen, where we inactivate brain endothelial cell exclusively. So clearly we shut down endothelial cell by turning off NF-kappa B essential modulator, NEMO. So when we do that, so when we test for cognition in these mice, where we have inactivated endothelial cell, NEMO, these mice have chronic hypoperfusion, they have vascular pathology, and as you can see here, they have a variety of cognitive impairment, one being here the difficulty in nesting, so they don't build nest as well as the wild type animal. And when we ask them to interact socially, so normally a normal mouse will interact more with a new mouse than with a mouse with whom they are used to interact. This is totally impaired in the NEMO mice. But when we actually let the tamoxifen effect go away for 3 months, so the mice became totally normal, as you can see, they behaved very well in nesting, and they behaved very well as well in the interacting with the new stranger mouse. And looking into the pathology, our GAL3 signal, we were curious to see what would happen. And as you can see here, this is the control mice, NEMO mice, clear GAL3 positive into the internal capsule. And when they recover and have no more cognitive deficit, this signal is disappearing. So I think this is really something going on with the white matter that seems to actually occur concurrently. We don't really know yet what is the clear meaning, but there is concurrent happening of the two pathology. So I'd like maybe to conclude that we have cerebrovascular pathology in TGF mice that we know is not enough to create cognitive deficit until very, very late. But when we add another risk factor, we precipitate that. So we precipitate the microvascular rarefaction by the string vessel. Vascular fibrosis is already there and quite severe. We don't know if we have actually more microhemorrhage. We did not measure that. But clearly it results into hypoperfusion, altered hemostasis, white matter inflammation, oligodendrocyte maturation is altered, and that will lead to cognitive failure. So it seems that one risk factor seems to be something we can deal with, but when we have two factors, we can precipitate cognitive failure, and exercise and simvastatin are able actually to counteract quite a lot of that, and a lot at the level of the white matter. And I would like to close down on this slide that Liam did for her paper as the abstract slide. So physical exercise seems to be acting at every single level of the failures that we could detect in terms of cognition, vascular pathology, white matter pathology, microglial cell chewing myelin debris in the white matter, functionality of the action potential into the white matter. So, and simvastatin was also quite effective, maybe a little bit less in some of the tests that we looked at. So on that, I'd like to clearly thank the work of Lian and Shinkan, so both of them for their work. I also would like to thank Jessica, Maria, and Clotilde for performing some kind key experiment or for blinding the mice, because all the studies are done blind to the person doing them. So we only know at the end what is happening. So that's also quite a lot of work, blinding and analyzing the mice and reorganizing the group. And obviously the funding, I'd like to thank the funding of CCNA, the Alzheimer's Society, CIHR, and I'd like to thank you for your attention. And now the sun has been going down. So thank you very much for your attention.
Okay, thank you very much, Edith, for this fascinating talk. It was really great, and I think it's of great interest for a lot of us studying aging and the effect of exercise on aging here at Perform.
It's amazing what it does, I have to say, really.
Yeah,
yes.
It looks like, so that's also reassuring for all the study we're doing on human, of what's, like, what are your findings. So I will go over, we have a few questions so far. I think we have some time to go over most of those we have for the moment. So,
okay.
So I'll start by the beginning and we'll see. The first questions was at the very beginning of the talk, so I'm not very sure because the person used an acronym. Is perhaps you remember, I don't remember. To what degree is CI related to age or gender? but I'm not sure what CI is.
Well, VCI, I guess, if this is it, I mean, cognitive impairment, I believe. We didn't do male-female comparison, so that I don't look for sex, because we did in some previous studies on APP mice, on TGF mice alone, and we could never get a clear response. So I think sometimes the cohort were more impaired as male, sometimes the cohort as female were more impaired. So I cannot tell you that we have a clear, in that animal model, deficit that is sex-related. Regarding age, TGF high cholesterol diet mice, even the young adult, were getting impaired when we were feeding them high cholesterol. So it was not age dependent. I would say they probably run very early on at the reserved. And if you trigger them with the second risk factor, I think they collapse, whether as young adult or more elderly adult, because in our case, we see that in both groups. Yeah. But in terms of the vascular pathology, as I showed, it seems that the vessels are more resistant, a little bit more in the young, normal mice to cholesterol.
Okay. I'll go over it. Is that, yeah, I'll continue on the questions by order just for now. So there is a question on how did you tune what is a high cholesterol diet in your mouse model? Is that something you developed yourself?
Yeah, no, we actually took that from the literature. I think most of the paper we looked at were using between 1, 2, and 3% cholesterol diet to really do that. We selected 2, which seems to be an average. We measured the change in the plasma, total cholesterol, which seems to make sense that it was actually doubling. So I think it's totally classic in terms of the concentration that we have been using. Yeah.
The next question is probably difficult to answer on mice, but is the exercise hypothesis also valid for people with primary progressive aphasia?
Oh, progressive aphasia. I don't know for progressive aphasia, to tell you the truth, but I do know that a lot of work is being done with exercise, trying to recover cognitive function in human in a lot of pathologies, not only in Alzheimer's disease or in aging. So as I said, aphasia, I never really looked into that, but I would say it is beneficial in a lot of things. It's a very dirty drug. I think we have to realize that, and even for Alzheimer's disease now, we are not anymore looking for a clean drug, because the clean drugs don't work, it seems. So I think a drug that has multiple targets, inflammation, oxidative stress, the vasculature, maybe neurogenesis, most likely will have benefits. So I would not be surprised if there is work on that, actually. And even after stroke, people after stroke will benefit from exercise. So I would not be surprised that aphasia would also benefit from that. Yeah.
Our next question, we have a very interesting question on lactate in that concept. So we know that lactate is produced during exercise and it's transported to the brain. There is a link between lactate and recovery of memory deficit. So the question is there a relationship between those studies and your experiment? And is it possible that simvastatin and increase lactate by doing so and able to rescue the cognitive impairment?
Okay, that's an interesting question, because some people are looking for the exercise molecule. I had some group who wanted to collaborate with us because they had supposedly found the exercise molecule, and I think lactate is one of those. Now, lactate will surely go into the brain, will be incorporated into the cycle to provide energy to the neurons, and so I think it is clearly feeding the brain. Whether simvastatin, we did studies on simvastatin with FDG PET in APP mice, and APP mice have a decrease in FDG PET, so they have decrease in glucose uptake in the brain. Simvastatin was able to basically normalize that. So could that be then that simvastatin is acting by promoting lactate? It could very well be, because by promoting glucose, somehow you favorize, you favor the Krebs cycle, you favor the aerobic and the anaerobic metabolism of glucose. So it is a possibility, but I don't know of papers, we did not measure that, and I don't know of papers who would have measured lactate with simvastatin, but it may exist. But yeah, lactate will surely fuel the brain, and it will compensate for a metabolic need of the brain. Yeah, possibly.
Very nice. Our next question is about so you are showing what's the effect of simvastatin and exercise on TGF with high cholesterol. What would be the idea of using both of them at the same time? What would be the expected effect?
Yeah, that's a question I have, I think, every time. The problem is, I think the problem is different in human here and animal model. I think in animal model, once we fully rescue the memory, once we fully rescue the neurovascular coupling, once we fully rescue endothelial cell function, once we fully cool down GAL3 positive cells in the white matter, there is not much more we can do if we fully recover. So combining the two, I think we would not be able to quantify the benefit because we got basically 100% recovery. But I think in human, it is clearly a question that should be asked, because probably in human, in elderly patient or in patient with cognitive loss or cognitive decline, maybe we have to modulate the exercise that they can perform. So maybe they have lower exercise capacity, maybe supplementing then pharmacotherapy with some form of exercise. I think it's a totally valid question, but in the animal, that's something it is difficult to do because we rescue them, so we cannot make them super mice at some point. That I think is one of the limitations.
Do you think of using one as a primer to the other one, like starting with exercise and adding simvastatin?
Yeah, and I mean, we did simvastatin because we knew simvastatin was able to recover memory in Alzheimer's disease mice. We knew simvastatin was able to recover vascular problem in the TGF mice. But I think there are other drugs that could be used, I am sure of that could be implemented with exercise. But I really think, yes, whether you start with a drug and you add on exercise, or you start with exercise and you add on the drug, I think the drug that should be added may be depending on what is the cause of the pathology of that specific patient. So if he is hypertensive, maybe you add an antihypertensive drug, which also has pleiotropic effect, and which we didn't do that, but I'm quite sure that if we were to add a drug which has pleiotropic effect, antioxidant, anti-inflammatory, acting on neurogenesis, I'm quite sure we would also get a benefit, but we did not do it. So I think combination is something to contemplate for the human population. Yes. Yeah.
Okay. Our next question is about clarifications about your conclusions from the fact that when you have simvastatin and exercise, so the person understands that microglia and astrocytes activations reduced while cognitive impairments were rescued and CBF was improved, and is asking, his or her, I don't know, wouldn't an increased CBF make for astrocytic activity also increase?
Not why? No, I don't think that an increased cerebral blood flow would normally increase astrocytic. I think if I try to understand the question, what we found is that TGF mice, whether they're fed high cholesterol or not, they have astrogliosis. Okay, exercise or simvastatin is not able in the gray matter to reduce astrogliosis or microgliosis, but in the white matter, actually, they are both able to reduce those microglial GAL3 positive cells. Our hypothesis is that the pathology that we see for the cognitive decline is not in the gray matter, is in the white matter. So that's why I believe that the mice recovered cognition, because simvastatin was able not to act in the gray matter, but to act in the white matter. So this is what we believe. And this is why we were very, with the last model I showed you, the Nemo mice, that also showed cognitive impairment, we were very, very curious to see what would happen with the GAL3 in the white matter, and it was there. So I think the pathology for me looks, and it was gone when they recovered. So after we left the tamoxifen washout for 3 months, the mice didn't have cognitive impairment and didn't have any more GAL3 in the white matter. So I think the gray matter doesn't seem to be the primary tissue that we have to look at. It seems to be the white matter at that point. So even if exercise is good for astrogliosis and microglial cells in the gray matter, because exercise was able to rescue those, I don't think we regain memory because of that effect in the gray matter. Okay, but that's a hypothesis at that point, obviously.
Okay. I'll progress a bit. I will use this one. We observe in few studies that exercise reduce glucose consumption and increase lactate consumption, but CBF does not appear to be affected. Can you comment on the energy deficits regarding vascular pathology and exercise?
Well, that's interesting. So they say that when they do exercise, they don't have an increase in blood flow and they have a decrease in glucose metabolism. Is that correct, if I understand? Yes, and an increase in lactate. Huh?
Yeah, that's what I read.
Okay, that is interesting. I would say if I start with the blood flow, the effect of exercise on the blood flow, I think people have seen various effects. Sometimes they see no enhancement of blood flow with exercise. Sometimes they do see enhanced blood flow with exercise. In wild type animal that exercise, if they don't have a deficit in blood flow, we don't see an improvement of the normal blood flow response. So there has to have an impairment in order for us to see a rescue. For glucose metabolism, we did not measure glucose, as I said, in these mice. We did not measure lactate either. Again, it depends maybe of the level of intensity of exercise. Just as a comparison, the mice that exercise freely compared to the mice that we limited the exercise. If we look at neurogenesis, so neurogenesis was only increased in the mice with unlimited exercise, but not in the mice that had limited exercise. So could there be that depending of the regimen of exercise, of the intensity, that you have different effect on the glucose metabolism, how much the neuron or the glia to neuron connection for the metabolism is directed? I don't know, but maybe that's a possibility that there is different effect depending on the type of exercise, the intensity, the regimen. I don't know because we did not measure that at that point. Yeah.
Okay, I have a long question here. I will read it. I didn't have time to read before, so I'll discover with you. You mentioned earlier that lots of issues with Alzheimer's is due to vasoconstriction. When looking at exercise for treatment, could this statistically significant change be due to an enhancement in vasodilatations, along with triggering the mTOR pathway to cause growth within body like the neurogenesis or oligogenesis? Well, there is another part I will continue. Secondly, when using simvastatin for treatment, could the benefits viewed in your study be potentially due to the stimulation of vasodilatation by triggering the nitric oxide pathway to enhance vasodilatation? And lastly, you stated that simvastatin had a lot of benefits and antioxidant benefits, yet with any drugs, there are always side effects. So you potentially using a safer natural source of exogenous nitrates from dark leafy greens of beets be used to enhance blood flow and to the brain, and mainly the white matter help with all the issue mentioned without any potential side effects.
Okay, I think all of those points, I think it's true. All of that is true. If I start with simvastatin, yes, we know simvastatin can actually increase eNOS messenger RNA, can increase eNOS nitric oxide, endothelial nitric oxide synthase activity, will reestablish nitric oxide availability because it is an antioxidant, so it will remove the free radical that trap NO. So clearly, and we published on that in other groups as well. So clearly, I agree. Simvastatin actually is able to reinstate vasomotor function on its effect on nitric oxide. Absolutely true. And that is demonstrated, and it is true here as well. The benefit, do they act through NO? I think for the vasculature, partly, yes, simvastatin can actually, for the acetylcholine, it is probably acting through NO in part. It is acting also, we've shown that through the TRPV4 channel that are also involved into the response. So it's as I said, those drugs don't have a single effect, but for simvastatin, the eNOS is clearly an effect for the vasculature. Now, the natural source, obviously, I think, yes, the statins have side effects. Some patients do respond worse than others to these cramps and the side effect of the statin. So, would that be enough to only give them a something that mimics the NO pathway, or I think they are dirty drugs. You know, I know companies don't like that, but those pleiotropic effects are actually good. And I think if I would be hypertensive, hypercholesterolemic, I would ask my doctor to give me a drug that goes to my brain, because I would like to have those effects of antioxidant, anti-inflammatory, acting on nitric oxide, acting on a lot, neurogenesis. I would like to have those effects to protect my brain in addition to my cholesterol. So I think, I don't know, even natural medicine can be quite dirty. I don't think there is any single herb that will give only one effect. But yeah, I think that like exercise is probably the most dirty drug we can think about, but it is very effective. So yes, I don't know of the natural compound, but natural source of drugs clearly will be able to mimic a lot of these effects. I would agree with that. And the NO pathway, yes, for sure, absolutely. For the neuron, for the positive cognition, I can tell it's not enough to increase blood flow. We have drugs that we tested that can improve blood flow, can improve perfusion to the brain, can normalize endothelial function, but they will not normalize cognition. So I think, even though I was dreaming about that maybe 15 years ago, that by normalizing brain perfusion, we would be treating Alzheimer's disease or vascular dementia, I have to admit that our results in animals now show us it's not enough. So we have a drug, we need drugs that act on something more than the vasculature, it seems. Yeah. So here I think simvastatin and exercise act on neurogenesis or oligogenesis or maturation of the oligodendrocyte, which may be one part of the efficacy.
Okay, and we have a few last, and we will conclude with those. For the high cholesterol diet, what was the proportion of energy coming from fat? Would higher saturated fat diet have similar effect than this one?
Yeah, okay, that's a question that we had asked ourselves. We did not go to the high fat diet because we didn't want to go there. We kept the same diet, we only increased cholesterol. We didn't want to go to high fat because of all of the complication of high fat diet, getting multiple sources of energy and getting the animal fat. And we didn't want to get the mice to be fat, because mice that are getting fat on a high fat diet actually perform difficult in the cognitive testing. So it was very important for us to not increase the fat content of the diet, but only the cholesterol. Now, there is a group in Ottawa, the group of Dale Corbett is doing a lot of interesting studies on the Western diet or high fat diet. They feed the mice or the rat with hamburgers, hot dog, potato chips, whatever they can find which is fat. They become like balls because they're very big, but they have adapted some of the tests that they've done for cognition. And obviously, yes, high fat diet has also very severe effect on cognition. Absolutely, yeah. But you're more limited because if you put a high fat, very fat mouse in a pool, they swim. They don't swim, they float. So you cannot do testing. So I think most of the studies, if you don't want to study Western diet and obesity, will go for a pure high cholesterol diet, which is more clean in terms of cholesterol effect. Yeah.
While on the subject of exercise, would you think that the results you obtain regarding recovery or cognitive functions, cognitive impairment, could be related, so we mentioned that already, to the increased production of lactate, and also the link with memory, which could improve memory?
Yeah, the link with lactate, as I said, that's something we did not measure. So maybe that's a weakness. We did not measure it in the brain. We did not measure it anywhere. So I cannot, from these data, make the clear link. I can only base on the literature what is known. As I said, there are companies and people looking for the exercise molecule, and lactate has been one of those proposed molecules. So I cannot respond clearly because I didn't do the measurement with the lactate in our model. So was there a second part to the question that I forgot, maybe?
I think it's fine. So we have two last. So could characteristics of exercise such as intensity, in addition to the drug like simvastatin, be a factor potentiating the benefits for cognitive functions?
I think that, I mean, you have expert in your institute about exercise and, you know, high intensity exercise followed by less high intensity exercise. And I think that has been shown to be probably better than continuous type of high level exercise. So I think, yes, those, I am not the expert in that. I think, yes, you have a regimen of exercise that can be very well adapted and can be more effective. In the animal, the only thing I can say is that they get addicted. So they would just exercise the whole night, basically. They would get exhausted. So, and then when we said, okay, let's reduce the exercise to 2 hours to 3 hours, and it was as effective. So it is not how long the mice exercise and how intense they were exhausted. The benefit we were able to see them with the limited regimen of exercise as effective. It was as effective in recovering cognition than getting the mice to run 25 kilometers a night, basically.
We'll finish with a question from Habib. So in Alzheimer's disease, a topathy is observed affecting the neurons, but also the astrocytes. How to explain the benefits of physical exercise in this pathology when you have a topathy?
You're talking tau pathology, yeah. Well, I mean, let's see, you have data then, I suppose, to say that exercise has benefit in reducing tau pathology, if you're asking the question, and I suppose you do. I don't have these data. As I said, exercise is acting so broadly. I know that, I mean, and even in the Alzheimer's disease field, I don't recall a paper where they've done exercise in EPP mice and measure A-beta level, measure plaque, measure tau. So I don't know. If you have those data, I would like to be aware of that. But I think by improving vascular function, I would suspect it would improve clearance of A-beta, because A-beta is cleared through the vasculature in part. For tau, the hyperphosphorylation of tau, if it's reducing tau pathology, it may be acting in one of those pathways where tau is actually getting pathological. I don't have the intelligent response to your question, Habib. I'm sorry. No, I would like to. If you do have it, I'd like to be informed. If you know, yeah.
We will probably discuss that another time. I might have just a very last comment in general, because you have a very nice access to lots of information coming from different aspects of the physiology, and notably the astrocytes and the glia, which are difficult to address when you are working with human and standard neuroimaging techniques. So in general, how biased are we, or by missing all this important information in our standard analysis? Like where should we pay attention to or something?
For human studies?
When we deal with human study.
You know, we did run MRI studies in some of our mice, in the NEMO mice, the last study I showed you, where they have these GAL3 positive microglial cell appearing into the white matter. So we did DTI and we did FA into these mice with a group in Toronto, the group of John Sled. And we had the white matter track. It was clear that there were deficits in DTI and in FA, in the very, very small pyramidal tracks and all of those. So I think the white matter is, for me, at least in VCID, this is where I would look. I wouldn't look for a fancy marker. If you have some of your patients that exercise, can you follow a change in the quality of the white matter through MRI? I think that could be something to look at. This is where I would go with my animal, with my model. I would look at white matter change, and I think we were very surprised and very happy to see that by MRI we could see some changes. Now, would we be able to see changes by therapy? I don't know. That we have not done, because it was done in the NEMO mice and we did not do exercise in these mice. But I think, yeah, I think the white matter now, can you get more? Can GAL3 microglial cell in the white matter be labeled somehow more than by looking at DTI or FA? That I don't know. That I don't know.
Okay. So I think it's time, we're over time already. So it's time to close now. If you still have questions, feel free to contact Dr. Hamel directly, like to ask
Absolutely.
more
Yeah.
questions. And so I'd like to thank you again, Edith, for this really fascinating talk and excellent discussions we had on this important topic.
Okay, no, thank you very much. I'm very, very happy to have been there, and thank you for the invitation, actually, and thanks for all of you attending the presentation.
We had really great attendance. Thanks to everyone.
Okay, thank you, everybody, and stay safe. Bye-bye.
Bye.